All right, so if you just walked in, please hit the start of activity. And we'll move straight into results from one of the latest discussion board activities, which was something about the Duchenne muscular dystrophy frequency. And so you don't need to read all of these. These are just screenshots of comments that some of you made, all of which were making the same point. You found a paper that suggested that, DMD occurs in about 1 out of every 3,500 males. That seemed like it was a pretty easy fact to find, but then a lot of you commented things like, we were unable to find one article that stated how frequently women are affected. That was a recurrent theme, at least in all of these 5 responses. Like, I could find the male thing, but what about the female thing? Well, these papers also said it would be more rare. So the question is, since we're on this topic of Mendelian inheritance, why would it be more rare in women than in men?
Because women have an X chromosome that can compensate for the lack of dystrophin if they do.
So the Okay, so if one of these X chromosomes is wild type and one has the mutation, then would you predict this would be a dominant or recessive disorder? This is a recessive disorder. You don't have to predict it. The fact said, Duchenne muscular dystrophy is caused by a mutation that's recessive. So why is that a problem for males? If they, like women, they could have just one X chromosome that has a mutation, but they don't have the benefit of that second X chromosome, so it'd be sad for us. And so this individual would have muscular dystrophy where a homozygous female would be wild type. Of Duchenne muscular dystrophy is about 1 in every 3,500. So what is that really the frequency of? It's the frequency not only of males that have this mutation, but it also tells us something, a different frequency. It tells us about how common or how rare something genetic is. Not just the disease, but the cause. How rare is the mutation? How rare is the X chromosome that has the minus? Because the frequency of males that have disease, what else do they have? They have the chromosome, the X chromosome that has the Duchenne muscular dystrophy mutation. So for every one male that's affected, you know that he has one of those mutations. So if it's about 1 in every 3,500 males has Duchenne muscular dystrophy, that also means that about 1 in every 3,500 X chromosomes has that minus, the mutation, mutant version of that gene, dystrophin. So if that's the frequency of mutant X chromosomes, how frequently should we find women that have Duchenne muscular dystrophy? That is, what's the probability of what genotype?
X negative and X negative.
So mutant over mutant. What's the probability of getting 2 mutant X chromosomes by chance instead of just 1? Frequency of mutant X chromosomes is 1 in every 3,500 X chromosomes. What's the probability that by chance you get 2 mutant X chromosomes in one individual?
1 in 3,500 squared.
It's the frequency times the frequency, just like everything else we've done in this class that has to do with frequencies. You multiply the 2 independent frequencies. So the frequency of mutant X chromosomes is 1 in 3,500 times 1 in 3,500, which is approximately something like 1 in 10 million, if I do my math right. Fact check me on that if you want to. But that's the idea, is that, yeah, it will be much more rare in women than in men just because this is a recessive X-linked disorder. It's much harder to be a female and to get 2 mutant copies in general. So even though you couldn't find a citation that said that it was a particular frequency in females, I suspect that part of that fact was just based on math. We observed the frequency in males, we could calculate and predict how frequently it should show up in females. It was 1 in 12.2 million. So 10 million, close enough. Okay, so to give you practice, because a pedigree will definitely show up on the final, showing you one of the two pedigrees I posted at the slides at the end of last class that we didn't get to talk about. So I figured we'd take a little bit of time to walk through these, going one inheritance pattern at a time. We've got 5 different inheritance patterns, 6 actually, but one of them is, these are 2. Is this either Y-linked or cytoplasmic? What do we look for if it's Y-linked? If it's in males only. Is this in males only? No. So not Y-linked. There are some females that are affected. Females generally don't have Y chromosomes, so this is not Y-linked. What's the pattern for cytoplasmic?
It's not, because when a woman has a child with cytoplasmic disorder, every single child has it from the mother.
Okay, so in cytoplasmic in red, if a mother is affected, she will pass it to all of her kids, not just one, and fathers will never pass it to their kids. So this pedigree violates both of those because this father has passed this disease on to 2 of his kids, plus this mom did not pass it on to all of her kids. So right off the bat, these are the first 2 I would always suggest looking at first. Rule out Y-linked, rule out cytoplasmic. They're the easiest patterns. They're the most distinctive patterns to spot. And all you're looking for is any part of the pedigree that disobeys or breaks one of the rules for that inheritance pattern. So even though in this part of the pedigree, the mom is affected and passes it to all of her kids, right? That is a cytoplasmic pattern, but it's broken elsewhere in the tree. So you don't have to find out if every part of the tree follows the rule. You just have to find one part where a rule is broken, and that's enough to dismiss that as a type of inheritance pattern. So that means we go on to the harder ones. X-linked and dominant. The rule here, what's the rule? Pattern for a white X-linked dominant. If it's an affected male, how does he pass that trait on?
To any of his daughters.
It's dominant. He passes his X chromosome only to his daughters because he passes his Y chromosome only to his sons. So it should go from an affected male to all daughters. If it's X-linked and dominant. Is it?
No.
Okay, where's the rule broken? So we're looking for males that don't pass to a daughter. There's one right there. And from generation 1 to generation 2, that male should have passed it on to his daughter if this was X-linked dominant. Didn't, so it's not X-linked dominant. The rule has been broken. What about autosomal dominant? Does it look like a classical sort of dominant pattern? What's the main difference between dominant and recessive patterns usually?
Recessive tends to skip a generation.
Okay, so recessives tend to skip a generation. This one doesn't skip a generation, so that's maybe a clue that it's dominant, although we've seen exceptions where recessive occur in each generation. So this could also be recessive. We'll get there in a minute. So could it be autosomal dominant? What are the genotypes of these 2 individuals, the parents of the 3rd generation? Can you figure that out? So if this is dominant, what are their genotypes? Are these 2 individuals homozygous for the mutation or heterozygous for the mutation, if it's dominant?
Homozygous.
They could be either. They could be homozygous or they could be heterozygous. So if this is a dominant disease, and if that individual is homozygous, has 2 mutations, where did those 2 mutations have to come from? They had to inherit their mutations from someone, so who did they get them from?
One from dad.
From their from their parents.
Yeah.
So if this person is homozygous, then the parents would have to also be homozygous for a dominant mutation, because even just one mutation, if this is dominant, that individual, that mother, would be filled in. She has one mutation. If it's dominant, she would be affected, and that's the only way to get. So this is the case where if this individual was going to be homozygous like that, the mom would have to have at least one minus to pass on to the kid. The fact that mom is not affected, not filled in, means that she does not have a minus. Again, this is all assuming this is a dominant. We're trying to figure out, is this dominant? So if it's not if it's dominant and the mom's not affected, she has to be wild type, which means that her daughter has to have inherited a plus from her. So we can infer the genotype. We don't actually know whether dad's homozygous or heterozygous. It doesn't matter. We've figured out the genotype of the mom in generation 2. What about the dad in generation 2? What's his genotype? If this is autosomal and dominant. Same as mom. Yeah. This is the exact same cross, an unaffected parent with an affected parent having kids. So that genotype will follow the exact same pattern. That means that the dad in that case is homozygous if this is dominant, and that we don't know question mark over minus. We don't know what the affliction of that, what the genotype of that individual is, but that means that the son is also heterozygous. Now, why is it important in ruling out autosomal dominant, if we're trying to figure out if is this or is this not, why is it important to know the genotypes of those parents?
To figure out if it's a dominant or recessive trait.
Well, we're assuming, so this is how pedigree analysis works. Pick every single inheritance pattern and try to rule it out. So this is the time where we're saying, could this possibly be autosomal dominant? So we're assuming that it's dominant just for the purpose of this exercise. We do this for every different pattern until we figure out which patterns are possible, and that's going to be the answer. Well, let me ask you this. If we knew, I'm not saying that this is true, but if we knew that both of those individuals in the second generation were homozygous minus over minus, then what would their kids look like? If both of the parents are homozygous for the mutation,
All of them would be homozygous for the mutation.
all of them would have that disease, and we would know their genotypes too. But what if the two parents are heterozygous, which is what we just figured out? What would their kids look like?
There would be some that aren't affected by the mutation.
We would do the Punnett square of a heterozygote dad crossed to a heterozygote mom and discover that there should be a 3-to-1 ratio when we convert genotypes to phenotypes. Anybody with a minus is affected if this is dominant, and everybody that's got 2 pluses is going to be wild type. So 1 out of 4 kids should be unaffected, which means that if this is really autosomal dominant, and these really are the genotypes, they're both heterozygous, then out of 8 kids, how many should be unaffected? 2. So 1 in 4, 2 in 8. So we would expect maybe, if this is autosomal dominant, that maybe one of the daughters and one of the sons would be wild type. So is that enough evidence to rule out autosomal dominant as a possibility? Because these 2 parents are heterozygotes. They should have had, on average, 2 out of 8 kids being wild type.
You would have to do a chi-square to rule it out.
You would have to do a chi-square test to figure out if this is a significantly different pattern from expected. So we have 8 affected, 0 unaffected. And we're not going to do the chi-square test, but you know how to do it. Observed, expected. We've got affected, and we've got unaffected. So we observed 8 affected, 0 unaffected. We expected 6 and 2, that 3-to-1 ratio. And it turns out that this is not significant. So if you do the chi-square test, the p-value winds up being something which I'll tell you in just a second. Point 1. So it's close, but those observed and expected data are not significantly different. So what's our interpretation? Could this be autosomal dominant?
Yeah.
Yes. The observed and the expected values are not different. That means by chance this could be an autosomal dominant inheritance pattern. And it's just by chance that all 8 of their kids inherited the disease instead of 6 out of 8. So we can't rule it out. So autosomal dominant is a possibility. So what are we going to have to do if we're going to figure out if this is definitely the inheritance pattern? How would a geneticist do this? We've already ruled out Y-linked cytoplasmic X-dominant. It may be autosomal dominant, so what do we have to do? How many more inheritance patterns are there? Got autosomal recessive and X-linked recessive left. So we have to check those also. And if we rule both of those out, then this is the only option left, and then we can say, dear patient, this is an autosomal dominant inheritance pattern. But if we can't rule out one of the other two, then we don't come down to a single that has to be this pattern. We say, well, we don't know. It could be one of these 2 or these 3. So could it be recessive? Autosomal recessive or X-linked recessive? So it doesn't look recessive because it's in every generation. So what could we do to make this, again, this assumption, we're going to assume that it's autosomal recessive for this slide. So if this really is autosomal recessive, can we figure out how you could make this pedigree follow that pattern? In other words, how is it that this mutation passed from generation 1 to generation 2? Yeah.
Oh, sorry. The male, the affected male in generation 1 could be minus.
Has to be. If it's recessive.
Recessive has to be.
Okay, so everybody that's got this has to be minus over minus.
And then the unaffected female and male are carriers.
My sloppy version of half filled in symbols. Okay, so does that suffice? Is that enough to explain that this could be a recessive disease and follow this pattern? So individuals in the second generation can inherit one mutant allele from each of their parents, and then you would predict their offspring are going to, if these individuals are both homozygous for the mutation, because this is recessive, then they would, of course, pass it to all of their kids because all of their kids can only inherit minus alleles from their parents. So it doesn't look recessive because it doesn't skip a generation, but we could still come up with a way to make it work if we fill in the blanks. Any questions about that? So A recessive? Could be. I suppose we could do another statistical test, but we don't need to. Because if you have that cross, you'd expect half the kids to be mutant and half not. We get 2 out of 4 instead of 3 and 3. That's not statistically significantly different. It's the last day of instruction. People are going crazy. Okay, so last one. X-linked recessive. So far we haven't ruled out, what do we have? Autosomal dominant or autosomal recessive. So we're already down to, it could be one of two patterns. This is the last one. Could this be X-linked recessive?
No.
What's the rule for X-linked recessives? That's the one to start with.
If a male is carrying, he passes it to all of his daughters.
So the male is affected here, so he's minus over nothing, Y chromosome. So he's going to pass his X to all of his daughters. So that means his daughters, the affected ones, are minus over minus. Remember, this is going to be recessive, so they have to be minus over minus, which means unaffected daughters could be plus over minus. So mom could be plus over plus or carrier, we don't know. But in either of those ways, the daughter gets the minus from her dad and gets a plus from mom. So that fits X-linked recessive. It's the other side that doesn't work. Yeah?
How can the daughters be minus over minus if the mother is plus plus?
Oh, sorry. Good point.
So
So it has to
mother
be,
a carrier.
mother has to be a carrier. Thank you. Nobody's perfect. Especially this time of year. Okay, there we go. So yeah, that's how the daughters become possibly minus over minus. So an affected mom, we know that she's homozygous. If this is recessive and it's X-linked, both of her X chromosomes have minuses. So what does that part of the pedigree do that tells us whether this could or could not be X-linked recessive? Where does mom send her X chromosomes to? Which kids? All of them. So every kid down here gets a minus for sure from mom. What's dad's genotype? If this is X-linked recessive, this guy up here. He has to be, so male only has one X chromosome and he's not affected. So he has to be plus over, if this is X-linked, he's plus over nothing. He's got a normal X chromosome, that's why he's not affected by the disease, but he's got a Y chromosome. So he's going to pass his Y chromosome, his X chromosome, his Y chromosome to all of his sons, his X chromosome, which we know is plus now, to all of his daughters. So one of those genotypes does not match its phenotype in their kids. Who can spot it?
The male who, both males should be affected and both of them are not.
Right. That genotype, minus over Y, should be affected because the male's got an X chromosome, it's a mutant X chromosome. It had to be because it came from the mom who's homozygous with this mutation. So she had to pass the causing mutation to all of her sons, and there's a son that's not affected, so that rules out that it could be X recessive. That male would have to also be affected, filled in, if this is X recessive. So in the end, we come down to it's either autosomal dominant or autosomal recessive. Yet another case where there's not always one right answer. Sometimes there's a right answer, but it's got multiple parts. Any questions about pedigrees? So on the exam, I'll probably ask some simple questions, maybe a difficult question. I might, as some of you have asked, I might make multiple versions of questions and have you answer a certain number of them. You get to pick which ones to answer. We'll see. I haven't decided yet. I've still got time to make the exam. So we move on to what I hope will stimulate a little bit of discussion, more than usual, genetically modified organisms. So thank you for all of you that participated. I hope you found this much easier discussion board activity than the ones we have been doing, because you probably just sat there and thought, well, what doesn't have DNA in it? Chemicals, basically. Do you agree or disagree with any of these? Do all of these things not contain DNA? Or might some of them?
I mean, nasty water, like water that hasn't been
Fair enough.
purified, has DNA in it.
Okay, so anything that's an organism has DNA. So, sir. Anybody else? So chemicals, yeah, maybe.
I would say the milk and the alcoholic ones, like the dry wine or the plain alcohol.
Okay, so maybe milk. So milk is an organic product. So even if it's pasteurized or sterilized or filtered, might it still have some DNA in it because there was a cell or two from the cow's udder that got into the milk and the cell had DNA? Maybe. Vinegars are products of fermentation, wines are products of fermentation, so they had organisms in them at some point, and of course, the wine came from grapes, which were organisms that had DNA.
With the argument that milk isn't that, I would argue that olive oil doesn't have DNA in it because it has trace amounts of it.
Okay.
With the way it's
Processed.
It's processed.
It's pressed. What about egg whites?
I don't think so.
Isn't that all protein?
Right, so the nucleus of the oocyte isn't in the white, so it probably doesn't have DNA in it, but it did come from an organism that has DNA. So there's no right or wrong answer to any of this. It was just to start stimulating discussion about things like this. Although this is kind of an old example now, it's from 2015. There's a school in the Midwest, a university in the Midwest, that does sort of a consumer survey every year. And this year, one of the questions was whether or not foods containing DNA should mandatorily be labeled as containing DNA. And hence the title of the post, which was filed in, holy crap, WTF. 80% of Americans support mandatory labels on foods containing DNA. But that list you just came up with is probably the list of the only things in the grocery store that don't contain DNA.
Or may have trace amounts.
Or may have trace amounts of DNA. Yeah. So salt, definitely. Anything that's a chemical. And then we could also debate sugar. Sugar comes from plants. So how processed is it? Was it synthetic? Sucrose, dextrose, glucose, or was it extracted from plants? And might it have DNA? So anyway, that's a little bit of the... So that brings us to genetically modified organisms and the survey from the start of class. So let's take a quick look at your responses. So, should all foods that have been genetically modified, in your opinion, be labeled? Most of you say yes. Some of you say no. The next question was the DNA question. Should all foods containing DNA be labeled? And maybe I asked the question wrong, so if you answered no, please tell me why. Should it be mandatory that all foods containing DNA be labeled as such?
No, because most of your food would have a label saying this contains DNA. So like if people eat meat, it's gonna have DNA. If you eat plants, which I would assume most people at least would eat plants.
Or nuts, or yeah.
Yeah, that has DNA. So you'd be labeling a whole bunch of stuff that common sense says you should know that it has DNA.
Okay. So I should have asked a clearer question. I was also worried that some of you might have interpreted this as, should they be labeled with anything, like an ingredients list or a sticker that says what it is. So next year, I will write a better question. So fair point. So either you say maybe, either you say no, because everything would be labeled, it'd be a waste of money, or you say no, because everything pretty much has DNA in it already, and so what's the point of labeling it? Or maybe you say, yes, they should for a different reason. Was there anybody in here that has that opinion? Because it's worth talking about. This is one of the ways that we focus on in class where genetics meets society. Did I ask other questions? Is it possible that consuming a GMO could pose an immediate health risk to humans? Again, I probably should have written a better question, but let's see. About half and half. 56% think yes, could pose an immediate health risk to humans. What are those health risks? Yeah.
Isn't it like something, some foods that are genetically modified create, kind of have like a synthetic, or like they, like estrogen, like I think soy or tofu or something, has like, when it's genetically modified, for some reason it gets like a lot more estrogen, like synthetic estrogen. And for men, if they eat too much of that, it could be really bad for them.
I've heard that. I don't know if it's true, but yes. So that could be, right. So does genetic modification change the chemical profile of the food? And would that be harmful or helpful? Right? The next question asked, could it be that you should be able to modify foods to improve their nutritional value? Some of you said yes. But you never know what's, and I'm going to show you an example of that in a little bit. You never know what's going to happen when you start modifying foods. Then, of course, there's the, again, the poorly worded question. So there's the simpler answer, like, well, yeah, if I was eating a GMO potato and I didn't chew it enough, I could choke on it, which would be a case of a GMO posing immediate health risk to me, but that doesn't really get at the heart of the question. So then, yeah, in what situations could it be acceptable to genetically modify an organism? So most of you disagree about using it just to change aesthetic quality. And then there's maybe a little bit more agreement about quality of the product, nutritional value, disease resistance, increased growth rate, and maybe to eliminate pests. So you can genetically modify plants to produce chemicals that make insect pests less likely to eat those plants, for example. So let's work through a little bit of this in a different perspective. So the big question is, should we have genetically modified organisms? I shot this a few years ago. This is part of an advertisement I got from a store that's in our area. They're actually nationwide. And I would like to call your attention now. I'm going to give away my opinion on this just by underlining this, but take a look at the middle here. And the goal here is to come up with a quick definition of what does it mean to be genetically modified. So what is a genetically modified organism? What do you imagine when somebody says this thing that's at the grocery store has been genetically modified and you're deciding whether or not to buy it? Yeah. Okay, so if it's natural, if it's found in nature as that plant or animal, it hasn't been genetically modified, but anything beyond that we could call genetically modified. That's one view, yeah.
Well, the funny thing is they are technically describing a form of genetic modification because crossbreeding and selective, like basically selectively choosing like certain traits from breeding is genetically modifying. Like corn is genetically modified because we've chosen generation after generation to make it look a certain way.
So how would that fit with your opinion? Would something that's been selectively bred like corn?
Then there's something that some those causes or like human interactions as well. So that would still be modified.
Okay. So that may be one way to reconcile the two views, that if humans have been involved, that's genetic modification. Other thoughts about what it means to be genetically modified? What you see in your mind when somebody says this is a GMO food?
When you're purposely trying to do something with an item that gives you like a specific result, like you grab the tomato and you inject it with something so everything else just turns yellow along with it, instead of just doing it in its natural way.
Okay, so yeah, so I got two main things out of there. One is humans doing something for a specific purpose, and the other word you said was inject, which may be a common theme. Classes I've taught in the past, that's been a common theme, which is visioning like scientists in a laboratory doing something with a crop versus traditional breeding, which is what this is. Which, again, there's no definition. It's up to you. I'm just trying to provide alternative ways of thinking about GMOs. Because yes, to some, and especially to geneticists, crossbreeding and hybridizing is genetically modifying. It's changing the genotypes, it's changing the DNA that's present in an organism by breeding two genetically distinct organisms, even if they're from the same species. It's human intervention in food production. But it's not in the laboratory injecting stuff with things. But so the part that got me, of course, was the parenthetical piece at the end that says not genetically modifying, which is in sharp contrast to what they had immediately written, which was that there was selective breeding involved. But again, I'm not trying to say that you should think that this is genetically modifying. This is my definition of genetic modification. Any other thoughts about this so far? So unfortunately, I'm not going to get to the point where we decide whether or not any foods are or all GMOs are dangerous. Ultimately, most GMOs are not posing an immediate health hazard to humans. Some might be, and I will definitely show you one example of that before we end. So what's a GMO? Here are a few examples. I'll run through it pretty quickly. This is storytelling, not on the final, although the concepts might be, but the details of these stories aren't. So if you went out in nature, how would you know if what you were looking at was genetically modified? Right? Is this a product of genetic modification or the environment?
It's the environment.
It turned out, if you flip to the next slide, it's the environment, because you can buy these plastic boxes that you can grow squashes in, and they fill the cube, and then you take the plastic thing off, and you've got a square watermelon or a square pumpkin. But it's tough to know whether or not something you're looking at is genetically modified, and that's one of the points for should they be labeled. But if they're going to be labeled, then we're all going to have to come to a consensus on what the definition of a genetically modified organism is. Maybe there should be different stickers with different warning categories. Like this one was selectively bred, but this one was actually genetically modified in a laboratory. Speaking of doing things to organisms, a lot of, and again, this is where my opinion shines through. I'm very pro-GMO, but there's a lot of literature you can read about frankenfoods. Scientists are cloning things together, and that's, of course, what we've done with citrus, grapes, apples, and lots of fruits for years. Take citrus, you graft something that makes tasty fruit onto a rootstock that is disease resistant. So you plant the rootstock in the ground, and it provides disease resistance to the plant, but then you clone onto it a citrus that grows tasty Meyer lemons instead of those nasty natural acids or acidic fruits that don't taste good at all. Right. So are grapes genetically modified? Depends on your definition, but this is a hybrid. This is one genotype of grape on a different genotype rootstock. This really is a actually, Frankenstein was the doctor, not his monster. Sorry. It's a thing, but it's a Frankenplant. If you get it, then you know what I'm talking about. Inside joke. And then lots of traits, like fruit size, are controlled by lots of different genes. They're called polygenic traits. You don't just have a mutation at one gene at one spot that changes a huge tomato to a small tomato. This difference in fruit size was created by traditional breeding over the last millennium or less, maybe 500 years. And in this study, which you don't need to know the details of, they isolated a bunch of different genes. The product of all of them can contribute to this sort of variation in size. So just one of those mutations by itself was enough to change a fruit that size without the gene to a fruit that size with the plus, right? Not a very big difference. So what selective breeding does is farmers in the field or in the greenhouse or wherever find if you're trying to make a smaller fruit or a bigger fruit in this case, you find the variety, the random plants that make slightly bigger fruits, and then you use those as parents for the next generation. You keep you keep picking and growing the ones that are bigger. What's the genetic basis of that? Why do we have to do that process? Why do we have to have a field of tomatoes and go look through all the tomatoes and find the biggest one to mate with another big tomato to try to make even bigger tomatoes? Why does it take so long?
Well, for instance, if big tomatoes is a recessive gene, then you have to find two of the tomatoes with recessive genes, and you have to breed those together, so then the rest of the tomatoes that you want to be big are all recessive, so they all are.
Bigger, yeah. So it can take a while to find the right parents. How long does it take for the mutation to crop up in nature that causes the tomato to be a little bit bigger or a little bit smaller? It depends on the mutation rate and the generation time of the plant. So if it's an annual plant, it takes a year to go from generation to generation. And if you grow a field of 10,000 plants, the mutation rate is 1 every 100 million nucleotides. It could take generations of humans, not just generations of plants, to find, by chance, those rare mutations that will make small changes in fruit size. It takes a really, really, really long time to do traditional breeding, which you probably already know or suspect. Because you have to wait until that random mutation occurs that gives you the trait that you're looking for. So then we've got this version. The only thing I want to say about this is a case of genetic engineering. So this is scientists actually changing nucleic acid sequence in an organism. And what they did was they turned up the amount of transcription of this gene. So it's the fish's own growth hormone gene. The scientists just found a way to make the organism produce more of it so it grows faster. So this would be an example of a cisgenic organism. It's been genetically modified in the laboratory, but no new genes have been added to it, and especially no genes from other species. They've taken the salmon's own DNA and just cranked up production of one of the proteins that was already being produced by the salmon. Good or bad? It's up to you. The point is, not every genetically modified organism necessarily is as bad as that sounds. So there's a spectrum. There's some, maybe, in my opinion, good GMOs and bad. Here's the best one. McDonald's wants to sell, you've been to McDonald's recently, you can get the kiddie meal with the little packet of pre-sliced apples. Maybe not. My daughters love to go to McDonald's, which is why we never go. But you can, right? So restaurants want to be able to package sliced apples and not have them turn brown and nasty. So this company in Washington State, again, cisgenesis, turned down, by mutating the DNA of the apple purposefully, they turned down the amount of this enzyme that causes apple browning when the apples are cut so that you can package the apples and they don't brown as quickly. Right? It sounds like most of you are probably against this because this is one of those aesthetic mutations. It changes the visual appeal of the product. So for better or worse, that's a genetic modification. And then there's corn, the poster child of traditional breeding, which evolved from teosinte on the left, and it was domesticated in central Mexico a couple of millennia ago. So here's what the teosinte, the ancestral plant, looks like, and what we think of today as corn or maize, and what an F1 first-generation hybrid between them looks like. So they're totally different looking in size, color, nutritional content, and they don't actually look anything like the teosinte. It doesn't really look like corn. It looks somewhat like it, I guess. So here's what we know. It was, as I said, domesticated in central Mexico, and it was about 10,000 years ago or so. They found this shelter underneath this giant rock that had some tools that they could carbon date the tools, so they knew how old the tools were, and they found evidence of contemporary maize-like plant remnants on these stone tools. So that's how they figured out about how long ago modern maize or corn existed. Now, this is a genetics class, and we're talking about genetically modified organisms, but there's one more genetic-y thing we need to do in class, and that's to make a prediction. So the teosinte on the left, B, has these really, really, really hard coverings over the seeds, which is the part we like to eat. And so you could try to chew on that all you want, but you won't get anything nutritious out of it. There's a single mutation, a point mutation, that makes B look like C, where the glooms, the coverings of the seeds, open up a little bit, giving humans access. So that one mutation made a really important change in the ability for humans to use teosinte as a food source and then start growing it and crop improving it, growing it bigger, larger ears, tassels that are easy to just come right off the cob without having to like pick every one off. So there's one mutation that makes B look like C. So here's what the scientists did to figure out exactly what this first random mutation that probably took eons, millennia maybe, to show up to help humans start using this as a food source. They took however many different individual strains of corn. Think of those as like different seed packets you'd buy for different types of corn, sweet corn, yellow corn, white corn. They buy a bunch of different types of corn and they DNA sequence different positions. Wait, oh, here we go. Negative numbers. What are negative numbers? Negative 1016, negative 410, negative 1009. This is the promoter of a gene, different parts, not all consecutive nucleotides. And then down here we get to minus 98, minus 90, minus 64, minus 14, and 18. So we've got one nucleotide here that's in the coding region of whatever gene this is. And then they also took a bunch of naturally existing teosinte strains, and they sequenced, again, whatever gene this is, to be revealed on the next slide. And what they're looking for, this is genetic mapping, they're looking for a mutation that distinguishes all of the maize, one genetic difference that distinguishes all the maize from all of the teosinte. And they don't find a single spot in this promoter of the gene, but there is a spot in the coding region where every individual corn plant has a C. The dot means it's the same as the letter above it, the period in each of those rows. So that's all C. If you look at that nucleotide in that gene, if it's corn, it's got a C. If it's any one of these naturally occurring teosinte plants, every single one of them had a G. This is correlation, but this is what geneticists do to try to find the mutations that underlie phenotypic changes. You collect a bunch of individuals, you ask what's common to all the ones that have this phenotype and what's common to all the ones that have this phenotype. So, last Socratic of the year. And the last question. So I just launched that, so you should be able to get in, but here's the setup for the question. This gene is called TGA, for teosinte gloom architecture. That's the name of the gene that they're looking at. And they know that, here's that G and C again. All of the teosintes had a G at position 18. We just looked at that on the previous slide. Nucleotide 18 is a G in teosinte. It's a C in maize. And there's a single amino acid difference that is caused by this point mutation. Teosinte has lysine, or K, and maize has asparagine, or N. So the question is, based on the codon table, which of the three reading frames, is it 16, 17, 18 that's a codon? Or is it 17, 18, 19 that's a codon? Or is it 18, 19, and 20 that are a codon? And that's the question. So where in the codon table are lysine and asparagine, and how would a G to C mutation cause an asparagine or a lysine to become an asparagine? So where in this table is a G to C change cause a lysine to become an asparagine? So which position in the codon is this mutation? It's the third position. So if we go What was the mutation again? An A to a C, C to G? G to C. So if that G mutates into a C, then you go from lysine to asparagine, and that means the reading frame is AA, and then ends at that third nucleotide where the mutation is. So that would be the first choice, 16 through 18. That mutation happens at the third position in the codon. And that one mutation causes genetic modification of teosinte to become a little bit more like corn. So that is a mutation, but one SNP causes that change. Oh, I was going to tell you the story about potatoes. If you want, I will tell you later. That was the example of the one not safe conventionally bred plant. The short version is farmers were trying to make a potato that made better potato chips and accidentally made it produce more toxin. So they selected for one trait, makes nice golden crisp potato chips, but it actually makes like 3 or 4 times as much of a toxin that potatoes normally produce, but it was a lethal amount. So somebody luckily fed it to a rat before they put it into production, and the rat died, and then they decided not to use that potato in production. So just because it's traditionally bred does not mean that it's safe. And just because it's mutated in the laboratory doesn't necessarily mean it's bad. Thanks for your attention this term. It's been fun. I will see you Monday or not. If not, have a great break.